Hello students, this is Vivid Botany. We are seeing a question about the cell cycle here. We know cell cycle includes the interphase as well as the cell dividing stage, and in between two cell dividing stages like M phases, like mitotic phases, there will be another phase called as the resting phase. So in this resting phase that we see here, the interface is divisible into three phases that includes here the G1, S and G2. G1 is gap 1 phase, S is synthetic phase and G2 is the gap 2 phase. So during these phases, let us see what happens. In the G1 phase, that is gap 1 phase, we find here the RNA protein synthesis will be taking place because that is very much required for cell division. Cell organelles are newly formed like chloroplast and mitochondria. We know chloroplast mitochondria are the semi-autonomous cell organelles. They contain their own DNA, so they undergo replication on their own without dependence on the nucleus of the cell. So those chloroplast and mitochondria multiplication takes place during the G1. That I can say that cytoplasmic DNA replication takes place during the G1 phase. And cells attains maximum size, that the maximum growth of the cytoplasm or you say protoplasm of the cell takes place, due to which the cell will become preparatory for the next coming cell division. And one more interesting feature here is that the cells which do not undergo any cell division further, the cells which do not undergo any cell division further, like heart cells, I mean the neuron cells, the cells of the brain, and permanent cells like in case of phloem, xylem, those cells which do not undergo any further cell division, they exit the cell cycle. They exit the end of the G1 phase and they enter into some resting phase that is called as quiescent stage. Quiescent stage, and during the quiescent stage, the cells remain metabolically active, but further they do not undergo any kind of cell division. That is the most important one. The cells which do not undergo further any cell division, they exit at the end of G1 to enter into G0, called as quiescent stage. Next, S phase or synthetic phase, and the synthetic phase is also one of the longest durated. Here the nuclear DNA synthesis takes place, right? The amount of DNA will get doubled. For example, if it is 4 picograms, it becomes 8 picograms, and number of chromosomes remain same, right? Remember, the two most important things is that the amount of DNA gets doubled, but number of chromosomes will not double, they remains same. For example, if the DNA amount is 4 picograms, it becomes 8 picograms, but the chromosomes, if they are 46, they remain same as 46, they do not will become 92 like that. So it is the most important thing that takes place in the S phase, that is synthetic phase. Now the next it is gap 2 phase, G2 phase, where tubulin protein synthesis takes place. Tubulin protein, we know it is very much required for formation of spindle apparatus, and spindle apparatus are required for karyokinesis, that is division of nucleus, pulling of chromosomes towards opposite poles. So that takes place during the G2, and cell growth continues even during the G2 also. Another important feature of synthetic phase is that histone protein synthesis takes place during the S phase. We know histone proteins are basic proteins, they are synthesized by basic amino acids like lysine and arginine, and these histone proteins are present in nucleosome. We know that H2A, H2B, H3, H4. There are four kinds of histone proteins occur, two molecules in each type. Total, a nucleosome is made up of an octamer. And around the nucleosome, the DNA coils twice to form the superhelix. Out of the nucleosome, we find another protein that is called as H1 protein. Along with H1 protein, a nucleosome is said to be made up of 9 proteins that are basic proteins. DNA is acidic, so it is negative. We know histone proteins are basic, they are positive. So, the acidic nature, the negative nature of DNA gets neutralized, balanced by the positive nature of the histone proteins. That is how the DNA gets electric neutrality and chemical inertia, that is chemical unreactivity, because it has to act like the genetic material. Now, let us see the question here, the cell cycle. In this cell cycle, what it is asked that some dividing cells exit cell cycle and enter vegetative inactive stage called Quiscent stage, that is G0 stage. This process occurs at the end of, that is M phase, G1, S phase, G2 phase. So this process takes place in G1 phase, that is at the end of G1 phase. So the cells which are not going to divide, which are not needed to undergo further any cell division, these kind of cells will exit at the end of G1 to enter into a quiescent stage called as G0. Right? So this is the answer here. Answer to this question is quite interesting. Very nice question. It's been asked from cell cycle, that is +1 biology. Hope you are able to understand my explanation. You have any queries, any doubts, please put up that opinion in your in comment box of my channel, and don't forget to subscribe my channel, that is Vivid Botany. We will be expanding our videos to even chapters also very soon. I will be explaining one chapter after the other in a series manner, so please do stick to my channel and widen your knowledge, widen your scope of success in NEET, the one and only competitive exam for medical aspirants. So thank you very much. Thank you.